Picture this. You're a marine biologist in 1922, pulling up your deep-sea net after hours of trawling the darkest parts of the Atlantic Ocean. And as you sort through your catch, you notice something strange. Female anglerfish, those nightmare-fuel creatures with the glowing lures and teeth that would make a dentist weep, keep showing up with these weird fleshy growths attached to their bodies. Little parasitic-looking lumps. And so naturally, being a scientist, you catalog these parasites, preserve them in formaldehyde, and move on with your life. Except here's the thing. Those weren't parasites. Those were males. Yeah, you heard that right. The males. And what you're looking at isn't some disease. It's reproduction. Anglerfish reproduction, which might just be the most disturbing reproductive strategy in the entire animal kingdom. And before you think, okay, so the males are smaller, big deal, let me give you some actual numbers. In the species Photocoronus spinoceps, females grow up to 46 millimeters, while males max out at 6.2 millimeters. But in Serratius holbwelli, it gets absolutely insane. Females can reach 119 centimeters, over 3.9 feet, while males are barely 14 centimeters. That's a 60-to-1 size difference. To put that in human terms, if a woman was 5'6", her male equivalent would be 1.1 inches tall. And if that isn't disturbing enough, the male doesn't just stay small. His body literally fuses at the tissue level with the female. His blood vessels merge with hers. His organs dissolve, and he stays there for the rest of his life as what scientists call a sexual parasite. And the record holder, a female collected in 1912, now at the British Museum, with 8 males attached. 8 separate organisms fused to one female. In 1925, Icelandic biologist Bjarni Samuelsson was examining preserved anglerfish specimens and noticed something odd about those supposed parasites. They had vertebral columns, spines, fish bones. He published his findings suggesting these might be dwarf male anglerfish, but the scientific community was skeptical. And in 1926, British ichthyologist Charles Tate Regan confirmed it. These growths were male anglerfish physically fused to females. But the real breakthrough came in 1975 when researcher Theodore Peach from the University of Washington published a comprehensive study examining over 100 specimens, found females with up to 8 males attached, documented males at various degeneration stages, and revealed that this was their primary reproductive strategy for at least 23 species. And scientists didn't figure out why this didn't kill both fish until 2020. That's right, 2020. Nearly 100 years after discovering the phenomenon, to understand why this evolved, you need to understand where these fish live. Serratioid anglerfish inhabit depths of 1,000 to 4,000 meters. At 1,000 meters, pressure is 100 atmospheres. That's 1,470 pounds per square inch. Temperature hovers around 2 to 4 degrees Celsius, just above freezing, and there's zero sunlight, complete darkness. But the real problem is food. A 1,980 study by marine ecologist Michael Angel calculated that biomass at these depths is 100 to 1,000 times lower than surface waters. At 2,000 meters, there might be only 1 to 5 grams of living organisms per cubic meter of water. That's 1 teaspoon of life in a space the size of a telephone booth. And finding a mate? A 2011 mathematical study calculated the probability. Given population densities and how far males can search, encounter probability is 1 to 10%. 9 out of 10 males die without ever finding a female. So how do males find females in pitch black water across vast distances? Pheromones. But the mechanism is insane. A 2012 study found that male anglerfish have olfactory organs 3 to 5 times larger than females relative to body size. Their olfactory tissue contains 50,000 to 100,000 sensory neurons per square millimeter. For comparison, dogs have about 25,000, and these neurons can detect pheromones at concentrations as low as 1 part per trillion. That's like detecting a single drop of water in 20 Olympic-sized swimming pools. Males follow these chemical trails through complete darkness, swimming for potentially weeks, racing against starvation, because here's the nightmare part. In many species, males don't have functional digestive systems. They're born with a countdown timer measuring how long until they starve. Their entire life goal is find a female and fuse before you die. That's it. And when they finally find a female, they don't court her, they bite her, and they don't let go When the male bites, his teeth release enzymes. A 2017 study identified 3 enzyme families in male saliva: metalloproteinases that break down collagen, hyaluronidases that dissolve the gel between cells, and fibrinolytic enzymes that prevent clotting. These enzymes liquefy tissue at the contact point, allowing the male's mouth to sink into the female's body. But here's the crazy part. The female's body responds by releasing its own growth factors. Within hours, she upregulates production of VEGF, vascular endothelial growth factor, by 300%. This causes blood vessels from both organisms to grow toward each other through the liquefied tissue at rates of 50 micrometers per hour. Within 48 to 72 hours, the vessels connect. The male's blood flows into the female's body and vice versa. They now share a circulatory system, and then the male's body begins to die. Researchers documented exactly what happens to males after fusion by examining specimens at different stages. Week 1 to 2, the male's mouth fully fuses, teeth dissolve, jaw bones resorb, eyes start degenerating. Month 1 to 3, digestive organs atrophy. The stomach shrinks to 10% of original size. Intestines collapse. The brain begins dying, with sensory processing areas going first. Month 3 to 6, external features disappear. Fins become nubs. Skin pigmentation fades. Month 6 to 12, the heart may stop beating entirely. Kidneys reduce to minimal function. After 12 months, what remains is essentially just testes, a thin layer of skin, and blood vessels. The male has no brain activity beyond basic autonomic function. He's a biological automaton, a living sperm bank attached to the female's body.
So how does the female use these attached males? A 2013 hormone study revealed the mechanism. When females are ready to spawn, they release gonadotropin-releasing hormone into their bloodstream. Because the circulatory systems are fused, this hormone reaches the male's testes through shared blood supply. The male's testes respond by releasing sperm. Within 2 to 4 hours of the female's hormone surge, males are releasing sperm. The female controls male reproduction through their shared bloodstream. She's using hormonal signals to remotely activate sperm production whenever she needs it. And females can live for decades. A 2010 study using otolith growth rings estimated some females were over 40 years old. If a female encounters a male every 5 to 10 years, she accumulates multiple males over her lifetime. And a 2014 DNA analysis found that multiple males on single females weren't related, meaning females are collecting males from across vast geographic areas. Genetic studies have pieced together when this evolved. A 2018 analysis found serratioid anglerfish split from other anglerfish 130 to 150 million years ago during the Cretaceous period, the age of dinosaurs. Sexual parasitism evolved 50 to 80 million years ago, and here's the fascinating part. It evolved independently at least 3 times, suggesting deep-sea pressures strongly favor this strategy. The extreme size differences came later, 30 to 50 million years ago, coinciding with ocean cooling that pushed populations into deeper, more food-scarce waters where males needed to be smaller and more energy efficient.